Hello, this is Dr. Ben Cisse, and this is Urban Biology. The focus for this video will be on protein synthesis. However, before you start watching this video, I strongly suggest that you pause right now, get yourself a box of colored pencils, get yourself pens, get yourself a pencil, but most importantly, also ensure that you have with you a notebook. This is a biology teaching video. Teach you in a way that you should be able to do very well in a high school biology class and also do well on the end of course test that is given by almost every state in the continental United States of America. All right? Okay, let's begin. So the goal for today is to learn about protein synthesis. So write the word protein synthesis. That is your goal. Your goal is to learn about protein synthesis, and I want you to underline the word synthesis. Anytime the word synthesis is used, what comes to mind? Some of you are familiar with the word photosynthesis. Okay? So the word synthesis actually means like putting together. All right? So now I want you to use one of your different colored pencils on the synthesis, and I want you to write the words putting together. All right? Write the words putting together. Okay? So protein synthesis means putting something together to make protein. All right? Let me repeat. Protein synthesis, it simply means that you're going to be putting something together to make protein. And that something you're going to be putting together is called amino acid. There are 20 different types of amino acid, okay? But in order to make protein, you can only use a selected amino acid as is detected by DNA, all right? So in other words, what am I saying? I'm saying that DNA has the code, the specific code to select a specific amino acid which you will combine together to make protein. So how does that work? In order for that to work, first we need to know what the structure is for DNA. All right? So go down one line in your notes and write the word structure of DNA. All right? So now we're going to look at the structure of DNA. Again, DNA is more or less like the principle, okay, of making protein. DNA is the master of making protein. So which means if DNA is not there, it is impossible to make the protein. But the question is, why is DNA the master? Why is DNA the principle for making protein? It's simply because in DNA you have genes. Genes are a section of DNA that holds the code for an amino acid. So the code dictates what amino acid from the 20 different amino acids, the gene holds the code to select one of the 20 different types of amino acids to put them together in order to make protein. All right? So now let's go. Let's look at the structure of DNA. So first what I want you to do is take one of your colored pencil and draw a circle. All right? Then with that same colored pencil, draw a slanted line. Okay? Now I want you to get another colored pencil and I want you to draw what looks like an hexagon. Okay? Then select another colored pencil and draw a rectangle. And then another color pencil and put your dot, dot, dot. All right? So what you see here is a simplified diagram of the DNA molecule. So when you look at this DNA molecule, you can pretty much tell there are three main parts of the DNA. First you have the circle, then you have what looks like a stop sign. That's the best way to try to remember it. A stop sign or an hexagon, and then you have a rectangle, and then you have a dot, dot, dot. Now, let's try to label the parts of this DNA structure, okay? The circle is called the phosphate. So write the word phosphate beside the circle. So the circle is a phosphate. The stop sign or the hexagon is the sugar. But this sugar is a special type of sugar called the deoxyribose sugar. Called the deoxyribose sugar. So yeah, this is sugar. But it's a special type of sugar that's called the deoxyribose sugar. We have the rectangle. The rectangle represents the different type of bases, nitrogen bases to be specific, that are found inside the DNA molecule. All right? I'm only representing one base here. However, I'm going to write the 4 bases that are typically found inside the rectangle. All right? So the 4 bases that are usually found in there, and I'm going to just write the initials of those bases for right now, and I'll explain later on what those initials or letters stand for. But this whole green structure right here is called the nitrogenous. It's called the nitrogenous bases. All right? So look very carefully. How many nitrogenous bases are there in DNA? Yes, if you say 4, you are correct. And what are the initials? At GC. Now the best way to try to remember this or try to recall it, think about where you are going or where you have been. You have been where? At GC. Where have you been? I have been at GC. So at GC are the 4 nitrogenous bases that are found in DNA. Now remember, you can't find all 4 of them in one of this block. There is only one that can be in each of the block. In this case, I'm only drawing one block. Okay, so in this one block, only one of these bases can be in that block. Okay, next we have dot dot dot. This dot dot dot, which is pink in my diagram, represents a weak hydrogen bond. How do you know it's weak? Because it's broken. It's not a straight line. So I'm trying to demonstrate that this is a weak hydrogen bond because it is what? It is dot dot dot. So let's label this as the weak hydrogen bond. Okay? The weak hydrogen bond. Well, that's the reason why this hydrogen bond is weak and not strong. Because on the other side of this DNA molecule is a replica of what you see on the left side. So there is a right side and there is a left side. So what you see on the left side is a replica of what happens or what you will see on the right side. Okay? And the left side and the right side are held together by this weak hydrogen bond. Okay? But why is it weak? The reason why it's weak because DNA has the ability to make an exact copy of itself in a process called replication. What is the process called by which DNA makes an exact copy of itself? It's called replication. Okay? So in the process of replication, guess what? The right side of DNA has to dislodge or separate from the right side. So let me repeat. In order for DNA replication to occur, the left side of a DNA molecule has to separate from the right side of the DNA molecule. And if that bond is too strong, that separation will not be easy at all. So that's why the hydrogen bond is weak, so that the left side of the DNA molecule will easily separate from the right side. Okay? All right, let's go over the structure again one more time. Our goal for today is to learn about protein synthesis. However, in order for us to learn about protein synthesis, we have to learn about the structure of DNA. But why? Why do we have to initially learn about the structure of DNA in order to understand protein synthesis? Because DNA is the boss. DNA is like the principal. DNA is like the master. DNA has the code for making protein. All right? So we need to learn initially about DNA. All right? Now, there are three main parts of a DNA molecule. First, we have the circle, which represents the phosphate. Next, we have the DNA. We have the deoxyribose sugar. All right, the deoxyribose sugar, which looks like a stop sign, or you can say it looks like a hexagon. And we also have the rectangle, which is made up of the bases. You can only have one base in this rectangle. So in this case, I have 1, 2, 3, 4 bases in there, but in actuality, it should have been 4 rectangles going down. But I'm just initially trying to let you know that there are 4 bases that are found inside a DNA molecule. All right? And of course, we have the left side of a DNA molecule, and then we have the right side of a DNA molecule. The right side and the left side are held together by a weak hydrogen bond. And I also explained to you why it is weak. It is weak because during the process of replication, during the process of when DNA is trying to make an exact copy of itself, the left side has to separate from the right side. So to make that process simple, to make it more easy, the bond, the hydrogen bond is weak, so that separation can go on easily. Okay? All right. So now what I'm going to do is, I'm going to draw again what I have on this board, but try to simplify it in a way that you can understand it. All right? So far we know that the DNA molecule is made up of those 3 parts, right? So these 3 parts all together, so take one of your colored pencil. So the 3 parts, these 3 parts can be collectively called the nucleotide. Okay? These 3 parts can be collectively called the nucleotide. So what's the nucleotide made up of? I'm going to just put it in parentheses. The nucleotide is made up of phosphate. Can you give me another structure that makes up the nucleotide? You are so correct. Deoxyribose sugar. Deoxyribose sugar. And last but not least would be the, you got it, nitrogen bases. Okay? Nitrogen bases. So the 3 parts that makes, so on the test, on the EOC test, they probably ask you what are the 3 parts that makes up, or all of these are parts of the DNA molecule except. So if they say all of these, then phosphate is part of it, the deoxyribose sugar is part of it, and also nitrogen base is part of it. However, if they ask you what the DNA molecule is made up of, and they only ask you a singular, or they need an answer, just one answer, then that answer will be the nucleotide. The nucleotide comprises of all of these 3 that I've written at the bottom. Are there any questions? I know I cannot answer you, but as I continue to videotape, I will be asking you questions. And I've been used to asking my students, are there any questions? So please don't laugh when I say, do you have any questions, okay? Hopefully this video will be able to answer your questions, okay? All right, so here we go. Let's continue. Okay, now look what's going to happen. DNA, I told you earlier, has 2 sides. So now let's draw the sides. So draw a circle, and then draw your house, like we did before. And then draw another circle, and then draw your house, like we did before. And then draw another circle. All right? So what you see there is a circle, a house, or a stop sign, and then a circle. Okay? A circle. The sugar, the circle is called a what? You're all right, phosphate. And then we have the house, which is the deoxyribose sugar, exactly right. So we have phosphate, we have sugar, we have phosphate, we have sugar, we have, did you see the trend? Do you see what is happening? They are alternating, okay? So the phosphate is alternating with the sugar, and the sugar is alternating with the phosphate. Okay, so keep that thought in your mind for right now. Okay, now use another colored pencil. Let's see what is going on. So you draw a line like this. I'm sorry. So let me use this color. Wait a minute. I'm sorry. Let me use this color pencil, the blue. Okay, there you go. So you got the blue and the blue. Did everyone see what I'm doing? Did you notice that the circle does not have any blue structure coming out of it? So the blue structure is only emanating from what? From the from the hexagon or from the sugar. Keep that thought in mind. All right. So now I'm going to draw dot 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 dot. Okay, so 2. Now remember, in real life, there are millions, okay, of these different 1, 2, 3 parts, 1, 2, 3 parts of the DNA molecule. But for clarity purposes, so this way you can understand it better, I'm only drawing a few here, okay? All right, so have that dot dot dot. So now this is the left side of the DNA molecule. So now I'm going to draw the right side of the DNA molecule. So on the right side, look what I'm doing. With your colored pencil, the same colored pencil you used to draw the rectangle, use that color pencil. So here we go. So you have the color pencil, okay? Now we are ready to draw another structure that attaches to this rectangle. What structure do you suppose will be attached to that rectangle? Think about it. Which structure? Is it the circle, or do you think it would be the stop sign or the sugar? You are so correct. It is the sugar, okay? So let's use that same color pencil we used. So there we go, circle, bam. There you go, so you draw your house again, that's your house. Seems like I drew it too close. So let me just get rid of this for just a minute. Let me just get rid of that. Okay, so let me space it out a little bit, and I expect you to space it as well. So that's this. So now you got the house, so that's the house. Then you draw, you got a circle, then you got your house. Then you draw, then you got a circle. Notice what is happening. Left side is a mirror image of the right side. Did you see that? Left side is a mirror image of what? Of the left side. What's on this side is the same as what on that side. Everyone got me? Okay, now let's join this together. So now we're going to join these, maybe the nitrogen bases that are not joined together. Okay? All right, stay with me. Please stay with me. Okay, now I want you to label this the inside of DNA. That's the inside. And of course, if this is the inside, then these two that's on the extreme on the other side are what we can say they are on the outside. Or write the word backbone. Okay, they mix up the backbone. Okay, so on the outside. So what I have written here could be written down here. Why? Because this side is a mirror image of that side. All right, are you ready? Look very carefully at this DNA molecule. Which two structures makes up the backbone of DNA? Look at it very carefully. What two structures makes up the backbone of DNA? You are so right. Two structures make up, this is the backbone. Backbone made up of what? Phosphate and sugar. Phosphate and sugar. But remember earlier we said that it's a trend taking place here. Notice that they alternate. So not only do we have the phosphate and the sugar making up the backbone of DNA, but they are alternating. You get it? So go down below this diagram. Go down below your diagram. I'm gonna go ahead and write it over here. But for you, I want you to go down your diagram, okay? And write down the backbone. Write down the backbone of DNA is made up of alternating, phosphate and sugar. Okay, please write this at the bottom of your diagram. Okay, right at the bottom of your diagram. So that's the backbone and that's the backbone. Okay, so technically I can say that DNA is made up of two strands. Or you will see this in biology books, they simply say made up of a double helix. Okay? All right, now let's go to the inside. What's the inside of DNA made up of? Remember what these blue things are called? Yes. So this blue, the inside, this blue altogether are called the nitrogen bases. Okay, nitrogen bases. So the inside is made up of what? Nitrogen bases. The outside is made up of what? Phosphate and sugar. Are the phosphate and sugar alternating? Phosphate, sugar, phosphate, sugar, phosphate, sugar? Yes. Okay? Now, let's concentrate on the inside of the DNA. Okay? Remember the 4 bases I told you comprise the DNA molecule? Yes. You have the A, you have the T, you have the G, and you have the C. Now, let's go ahead and I gave you the initials. Let's go ahead and get their full names now, okay? So we can try to understand what is happening in the center of the DNA molecule. The A stands for adenine. The T stands for thymine. The G stands for guanine. The C stands for cytosine. So these are the 4 bases that are found in the DNA molecule. Okay? Now, here's the tricky part. These 4 bases are couples for life. Let me repeat what I just said. These 4 bases are coupled for life. In other words, they are always in pairs. They will never be separated unless if there is what we call mutation. But if there's no mutation, if everything is going like expected, everything's okay, they stay together for life. Let's look at A. Who do you think A gonna marry to? Is A gonna marry to T, to G, or to C? Make a choice. Guess. What do you think? Yeah, A is gonna always stay with T, unless if there is, you're right, mutation. All right. So now let's go to the box. So if I have A in this box, T always going to be in that box. Why? Because they are married for life. They are couples for life, happily married for life. They will never be separated. Oh, tell me. Easy part. Who is G going to marry to? Guanine. G is going to marry to cytosine. Great job. So you put a G over here and you put a C over here. Look. Don't get confused, okay? I can write A over here and also a T over here. It does not mean that A always have to be here. It can be either or. Either is in this box or in that box. But the bottom line is those two always go together. They always marry together. Who's A always married to? T. Who's G always married to? C. Who's A always married to? T. Who's G always married to? C. Exactly right. They are couples for life. You got me? All right. So what you see here is the structure of a DNA molecule. All right. So now, why is the DNA molecule important for protein synthesis? That's a question now we're going to tackle. Are you ready? I am ready. I am excited. And I hope you're also excited. Okay? So let's go. Okay, now, write this down. How does DNA make protein? How does DNA make protein? We already know about this DNA, this all-powerful DNA. We already know the parts that this DNA is made up of. Now, how does the DNA now make the protein? So in order for me to explain how DNA makes protein, I'm going to draw a diagram, or we are going to draw a diagram. So I'll draw and you draw. If I say use a certain color pencil, select a certain color pencil. It does not necessarily mean that you have to use the same colored pencil I'm using. Okay? Are you ready? Here we go. Okay? All right. So make sure you have a clean page. There is nothing written except how does DNA make protein? Or how does protein synthesis occur? All right? So it's a clean page. Now I want you to take your page and go down halfway of your page and then go all the way to the margin. So you select blank at the top, blank at the bottom, go about halfway down your page. Okay, halfway. Now, I want you to select one of your colored pencils. Okay? Select one of your colored pencils and draw what looks like a circle, but in reality, it's not a real round circle. Okay? But for explanation purposes, I want you to draw a circle with one of your colored pencils. Okay? All right. With that colored pencil, I want you to put in dots. So let's use another colored pencil to put in dots. So put dots. These are pores. Pore. Pore is like a hole. Okay, that's a pore. That's a pore. That's a pore. So these are pores going all the way around. Pores going all the way around. Okay? Those pores are like holes that are found. Those pores are like holes that are found inside on the outside of a nucleus. So this circular thing that I drew is a nucleus. Don't write the word nucleus yet. Let's draw the diagram first. Then after that, we will make a legend to try to explain the parts of what is taking place. Okay? All right, so that's a nucleus. And those are the pores, okay? Now get you another colored pencil. Get you another colored pencil. So I'm going to draw this structure that looks like this. This is the symbol for DNA. The previous structure that I drew on the board is the three-dimensional, okay? So this one here is like a two-dimensional, I guess a good word to use. It is folded, so DNA is always like twisted. You know, that's why they call it a double helix. It's always twisted. So that's the DNA. So anytime you see the symbol, that is DNA. All right? Now let's go and use another colored pencil. If I'm going too fast, please pause. Okay? If I'm going too fast, pause. Okay, so get you another colored pencil. Okay, so with that colored pencil, I'm going to draw a short arrow, and then I'm going to draw just one wiggless, squiggly line. What structure is that? DNA. God, don't label yet. That's DNA's brother. Both of them have the same last name. You know what his brother's last name is? Nucleic acid. You know what his first name is? Ribose. Can you pronounce his full name? RNA, or ribose nucleic acid. RNA. So this is DNA, deoxyribose nucleic acid, and this is RNA, ribonucleic acid. Everyone get it? 1, 2. Okay, here we go. What are we trying to make? Again, what are we trying to make? Protein. Protein is made outside the nucleus, right here. That's why protein is made. Not inside the nucleus, but outside the nucleus. However, DNA holds that information for making the protein. All right? DNA, this is DNA, holds information to make the protein. But that's a problem. Look at the pores. This should be like the gate. One of these, either the RNA or the DNA, has to exit this gate to come outside the nucleus to make the protein. Look at it very carefully. Who do you think will exit that gate or that door? Do you think the nucleus can go through that door, or do you think the RNA can go through that door? Which one do you think? Try to guess. Which one do you think can go through? Yeah. If you say the DNA is too big to go through it, you are right. It's too big. Look at this. Double helix. Can't go through those pores. Those pores are too small. So you know what? DNA is smart. It said, okay, if I cannot go through those pores, listen, brother, why don't you carry that message for me? It said, I have the power. I have all the information on how to make protein. However, I'm just too big to go through the gate. Can you please take the information from me to the outside? And guess what guess what RNA said? Yes, brother, why not? I can go through the hole, but you can't. Okay? So now here we go. So DNA is going to give the message or the genetic code to RNA. So with the same color pencil or different color pencil, put a bracket around the nucleus. So what is happening here in the nucleus is what we call transcription. Transcription. So we have transcription taking place inside the nucleus. Why is it called transcription? Because DNA is transcribing the message or giving the coded message to RNA. Okay? All right. So now RNA has the message. What's it going to do with it? It's going to sneak through the door. All right? But remember, it's got the message, so think about it. It's taking the message somewhere. Okay? DNA specifically giving the instruction to RNA, said take that information to somewhere. We're going to see what that somewhere is. But for right now, use the same color pencil you use. That's the same color pencil. Okay, here we go. 1, 2, 3. RNA out. RNA out. RNA is going. RNA is going. RNA is going. And then it stops. So draw your RNA entire. So you the entire scan the entire paper all the way to the end. So RNA is going. But remember, RNA is not going aimlessly. RNA has a purpose. The purpose of RNA is to carry the message in the form of a code. All right? So who is RNA carrying it to? RNA is carrying a message to what we call the ribosome. What's RNA carrying? A message. Because RNA is carrying a message, it's no longer called RNA, now it's called mRNA. mRNA. The real name of RNA is ribonucleic acid. So it's capital letters, that's the name. But since it's doing a job by carrying a message, we write the message as a lowercase letter. So this RNA coming out is now called lowercase m RNA. Don't write that down yet. We're going to come to it later, okay? Right now it's just for information purposes only, okay? So who's RNA going to? So go to your next color. Go to your next color. Quickly go to your next color and let's see where RNA is going to. So the mRNA now is going to this double lobe that looks like this. This double lobe is called the ribosome. Where is mRNA going? To the ribosome. DNA says, take that information to the ribosome for me. So that's where it is going. It's going to the ribosome. Okay? All right. Now, we said mRNA is going. Look, man, look at that. It's plain, just like in the nucleus, plain. I don't see no message on it. So now let's put the message. Okay, are you ready? So for the message, let's use another different color pencil. Different color pencil. So now I'm going to put the message. The message is a 3-letter code. This is one message. 3 letters. 3 letters. 3 letters. 3 letters. And then the first letter is always inside the ribosome. Look, 1, 2, 3, 4, 5. Those are messages. Obviously, in reality, there are millions. Okay? Depending on what DNA wants. But again, this is just for informational purposes. Are you with me so far? Good job. Okay? So these are the messages. This message are called codon. So this is codon, that's a codon, that's a codon, that's a codon, that's a codon, but that's a specific codon. This also is a codon, that's a specific codon. The codon that always begins information or gives information how to make protein is always called the start codon, and it always begins with AUG. AUG. That's always the start. And obviously, if this is start, let me write the word start over here. So that's where it starts making the protein. Obviously, you can't continue making protein forever. You can't keep on going on and on and on and on and on. There will come a point of time where you have to stop. So therefore, the last one is called the stop codon. It tells it, DNA is saying, I want you to combine millions of this code. However, you have to stop after that million is over with. That's it. So you have to stop coding. Okay? Okay. So now mRNA brings the message from out of the nucleus to what we call the cytoplasm. So everything outside the nucleus now is called the cytoplasm. So what is next? Now ribosome going to read the message. First, it's going to read this first one, which is called the AUG. That's the first codon. And when ribosome reads that message, it's given information to select amino acid that's scattered all over the cytoplasm. But I don't see no amino acid, do you? I mean, it's kind of plain there, isn't it? There's no amino acid. So let's go ahead and put the amino acid. Okay, there are 20 different types of amino acid. So with your next color pencil, let's put in amino acid. That's an amino acid, amino acid, amino acid, amino acid, amino acid. Let's put all of this down at the bottom for right now. They all also at the top, but just put them at the bottom. So that's amino. So there are 20 different types of amino acid. All of these blocks, what looks like rectangle, all of them are amino acid. Are you with me so far? Okay, so now ribosome reads the message, and then somebody gonna pick up that correct amino acid and bring it to the ribosome before the ribosome will move to the next code. Okay, that's somebody that's gonna pick that correct amino acid with another colored pencil. That somebody looks like a rake. So draw, looks like a rake, look like a rake. So each of these right there carry their own specific amino acid. I'm just going to draw 3 or 4 or 5 on there. So all of them have their own specific amino acid to carry. Once that information is given, once the ribosome reads that message is going to be relayed on to this structure that looks like a rake. That structure is called a tRNA. What's that structure called? tRNA. Don't write anything yet. We're going to label them later on, okay? So that's a tRNA. So guess what the tRNA does? It carries the correct amino acid based on the information it receives to the ribosome. Then the ribosome moves to the next code. And then the correct tRNA brings the other, okay? So first, let's see what is happening. So now go back to this diagram, and let's look at this diagram to that same color that you had right here. Let's look at this color right here. So look what is happening. Ribosome reads the message, and then it brings the correct amino acid. So that's the correct amino acid, okay, based on this code. So that's the correct amino acid. So ribosome moves to the next one, and then tRNA brings the next amino acid and joins it. And then the next. And then next. Until finally, what do you think is going to happen? It says what? Stop. That's it. That's all I need. Oh, by the way, what is this called now? These are all amino acid. When they are by themselves, it's amino acid. Listen, if I am standing here by myself, I am an individual. You got me? But if I am with other people around me, now we are in a group. I can't say I'm an individual by myself because I'm together with some other people. You got me? So if amino acid if this block is all by themselves, they are called amino acid. But once they join together, now they form something different that we call a protein. So this structure now is called a protein, or what we also call a polypeptide. Man, that's kind of hard to swallow, isn't it? This is basic biology, high school biology. It's not honors, but just simplified biology, questions that you will be asked on the end of course test. All right? So let's go quickly through it. Oh, by the way, forgot. Transcription takes place in the nucleus. Don't forget nucleus. This is the bracket. This bracket encloses only the blue, and that blue is the nucleus. Okay? Now, this is another bracket. Let's draw this bracket together. This another bracket. And that bracket starts from right here all the way across. That bracket is called translation. So that bracket is called translation. Okay? So that bracket is called translation. Okay? What's that bracket called? Translation. So let me ask you this. Based on this diagram, based on what you're looking at right now, how many steps are there for making protein? If you want to make protein, how many steps do you have to go through? Those 2. First, transcription. Second, translation. You get me? What's the first? What's the second? Translation. Okay? All right. Let's recap. Let's go over the whole thing again so you can get it. Okay? Are you ready? So now we're trying to make what? What are we trying to make? Protein. This is what we made. That's a protein. But how did we make the protein? You got to go through step. The very first step takes place inside the nucleus, and that's called what? Yeah. Who holds the information? DNA. By the way, that information is called gene. Gene is a section of DNA, and that gene has information to make protein. But the gene is in DNA. The DNA is holding that gene. Okay? So the DNA is much more bigger, it's much more fatter, like me, and the RNA is slimmer. Why is that important? Because they have to go through this hole in order to make protein, because the protein is not made inside the nucleus. Protein is made outside the nucleus. Even with me. Okay? So who's going to come out? Is it the DNA or the RNA? You got it. The RNA is coming out. When the RNA comes out, where is the RNA going? That's the RNA going. Yeah. Going. It's got a purpose. It's got a reason for going somewhere. It's not just going aimlessly. The RNA knows exactly where it's going. Where's it going? To the ribosome. Okay, the snowman over here is the ribosome. That's the snowman, it's the ribosome. That's where RNA is going. Wait a minute. When RNA is going to the ribosome, is RNA carrying anything? You got it again? Exactly right. RNA is carrying the genetic code. It's carrying the code. How many bases make the code? Count. 1, 2, 3. That's one code. 1, 2, 3. So how many bases make a code? 3. Okay? Now, once RNA brings the code, now we're going to translate the message. You got to interpret the message that came from DNA. All right? Who's going to translate it? 2 important structures are involved. Ribosome, tRNA are involved. Okay? All right. So ribosome reads the message, sends the information to tRNA. tRNA picks the correct amino acid, brings it to the ribosome, and then puts them together, and bingo, you have a protein. Okay, now let's go to our legend. Okay? Legend means the colors that we use now, we're going to describe what happens to all of those colors, okay? Let's go to those colors. Okay, first, this is mine. Let's go to the blue, walking away from left to right. So the blue that I use, the blue, okay, the blue that I use, that's the nucleus. So I'm going to label that the nucleus. That's the blue, okay? Blue is the nucleus. Then I use the red. The red that is coming out, to be specific, we call it lowercase m, R, N, A. All right, so that's the red. Okay, next you have the, let me see what color. Let's go to the black. So my black, I'm not sure what color you use, but mine is black. So that right there is that's the ribosome. So that's the ribosome. So make your legend, ribosome. Okay, that's the ribosome. Okay, next we have the green. We have the green. Okay, that's the green. So that's my green. That green is called the amino acid. Okay? That green is called amino acid. And like we clarified earlier, there are 20 different amino acids. Okay? Then we have the code. There are 3 codes, 3 of them. These are called codons, okay? So the 3 codes, we call them codons. So those are the codons. All right? Those are the codons. All right? Okay, what other color? Yeah, let's look at the brown. So let's look at the brown. So the brown, I'm going to come all the way over here, make my legend on this side, make it look nice and neat. It's lowercase t, r, and a. All right? So that's the brown. Let me see, am I missing any other colors? Any other color? Okay, let's go back to the green. When you go to the green, if you have more than one, they are attached together. Now you don't call it amino acid anymore. Now you can call it protein. Another name is called peptide. Okay, so the green, if they attach, they call protein or polypeptide. All right. Okay, next. Okay, I think we have 1, 2, 3, 4, 5, 6, 7 colors. I think we have used up, DNA. So we got the purple. I'm sorry, almost, thank you for reminding me. I heard someone say you forgot DNA. That's right, DNA. So DNA will be like this. So that will be the master of all of this. That's the DNA. And inside the DNA, you have what you call genes. Okay? And of course, the nuclear pores, don't worry about those. The nuclear pores are fine. So those are your legend right there. All right? Those are your legend. And the legend tells you what each of those colors represent. All right? Now, we are ready to explain what each of them does. Okay? We're ready to explain what each of them does. Now, I'm going to switch over, or I'm going to go to another screen. If you haven't finished, please just pause and then get as much information as needed. All right? Okay. So now, write the word protein synthesis. Protein synthesis summary. Okay? Write the word protein synthesis summary. First, go to the boss, the nucleus. Okay, so what's in the nucleus? The nucleus contains the genetic material DNA, contains the genetic material DNA, which holds the information for making protein. Okay, which holds the information for making protein. Okay. This is again what? Urban biology. We are teaching biology is and explained in a way that you can get it. So you can do very well in all of your biology classes, especially what we call the lower level biology classes, not honors class. Honors goes a little bit deeper, so this is more or less explained in a way that will help you be successful on the end of course test, like I said earlier, all right? So that's the nucleus, that's what the job does. So in the nucleus, you have the genetic material. You have ribosome not ribosome, you have RNA in there, you have DNA in there, all right? But the important thing you need to know for the nucleus, that it holds the genetic material, okay? Okay, next, let's go to B. Let's go to Mr. DNA himself. So what does DNA do? DNA controls the activity for making protein. Okay, DNA controls the activity for making protein. So why am I saying that? Well, because DNA has or contains genes. Make sure you write the word genes in caps or color pencil. Okay? So DNA controls the activities for making protein. But why DNA? Why not the ribosome? Why not mitochondria? Why not chloroplast? Why not other structures in the cell? Well, because the DNA contains genes. And genes has that information to give the code to make different types of, to combine different types of amino acids. Okay? Okay, next. Next you go to C. So now we go to lowercase m, R, and A. Lowercase m, R, and A. Lowercase m, R, and A. So what does the lowercase mRNA does? So this mRNA, I want you to underline that letter M. So basically it's a messenger. So this RNA carries the coded message. Write the word coded in caps. Okay, carries the coded message. carries the coded message to the carries the coded message called, carries the coded message called codon, okay, to the ribosome. So it carries the coded message called codon to the ribosome. That's its job. Okay? D. Let's go to its cousin. Its cousin is RNA. tRNA. As the name implies, the tRNA is responsible for transporting. So it transports the correct amino acid. Okay? Write the word amino acid in caps so you know exactly what the main purpose of the tRNA is. So the tRNA transports the correct amino acid. Where is it going? It also is going to the ribosome. So both brothers, okay, or cousins, brothers, cousins, whatever you want to call them, okay, both of them are going to where? Both of them are going to the ribosome. Okay? Okay, the next one, well, before we leave, there is something also important about the ribosome. How is the, not the ribosome, the tRNA. Why is the tRNA important in this process? Well, tRNA has what we call an anticodon. What the anticodon does is to read and break the code and select the correct amino acid. So this is what we drew before. So now this is tRNA. So on the tRNA you have the correct amino acid. Okay? On the tRNA you have the correct amino acid. So this is the tRNA itself. Now the structure may look a little bit different, all right, in books, but just remember there are 3 prongs at the bottom. Now these 3 prongs are called the anticodon. Okay? So the anticodon is going to break the code that is on the mRNA. So when you break the code, then it selects the correct amino acid and brings it to the ribosome. Okay? So just remember on the test, they will ask you which molecule has an anticodon. Okay? And that molecule is tRNA. Why? So this way it can this way it can carry the correct amino acid from the cytoplasm to the ribosome. All right? Okay, so let's go to E. So now we go to ribosome. Let's go to the ribosome. That's where all the action is going to take place. So most of you should know by now what the job of the ribosome is. Okay? What the job of the ribosome? So the ribosome, this is where amino acids, this is where amino acids are combined. Okay? Why are they combined? To make what we call a polypeptide, or we can simply say protein. So polypeptide and protein, the same. Simple definitions. Not too many worded definitions that get you confused. Simple and to the fact. Okay? So ribosome makes protein. How does it do? Combine amino acid. tRNA carries the correct amino acid. How does it do it? Has the anticodon to break the code. mRNA carries the message. What's the message called? Codon. Where is tRNA and mRNA both going? They're going to the ribosome. Okay? Do they have a reason to go to the ribosome? Of course. Why? To make protein. All right? Okay. The next would be codon. So let's go to F, codon. Okay, let's go to F, codon. So what does the codon do? This is a three letter basis that represents one amino acid. one amino acid. Okay? These are 3-letter bases that represent one amino acid. Since we're talking about RNA, let's say we have AUG. 1, 2, 3. This represents only one amino acid, and that amino acid is called MET, M-E-T. Okay? This is short. Actually, it's called methionine, but I'm just trying to shorten that word for you. So as you can see, when we say this represents 3-letter base, 1, 2, 3-letter base. This is the amino acid. That's A amino acid. And this right here would be the code. And how many bases make a code? 3. Okay? All right. We have 2 more definitions. 2 more definitions. I'm not sure what letter this is, I believe. Okay, 2 more definitions. I think this is, okay, this is F. So let's go to 2 more definitions. We have G. And that first one is called, the first one we started with called transcription. It's not transpiration. That's how plants lose water. But no, we're not talking about that. We're talking about transcription. Okay, transcription. Okay. What's transcription? This is what happens, all right? The transcription is what happens when DNA gives the coded message. This is what happens when DNA gives the coded message to mRNA. Where does that transaction take place? Nucleus. Okay? So that takes place in the nucleus. That's where that message is given, in the nucleus. And the last one. Man, I'm sweating, I'm tired. So this is the last one. That last one is H, which is called translation. Okay? Translation. Now remember, we're talking about a message. We're talking about a code. The message is not just a message. It's not like a verbal message. It's a code. So guess what? We've got to translate the message. Okay? So now you translate the message that came from DNA. But translate the message for what? Well, translating the message to make protein. Okay? So what's translation? This is where the coded message is interpreted in order to make protein. Okay? So the coded message is interpreted to make protein. Summarize. Don't be surprised if you see this on the EOC. Okay? Anytime you see a star, make sure you focus on the star. It could be on the EOC, all right? So what's the star? First, you start with DNA. That DNA is going to give the information to RNA, and then RNA, so forth and so on. There are 3 RNAs. You have mRNA, you have tRNA, then you have rRNA. Messenger RNA, tRNA, which is the transport RNA, and then you have the rRNA, which would be the ribosomal or ribosome RNA. Okay? So we have those 3 RNAs. So now what they're going to do, those 3 RNAs will group them all together as RNA, and all of them is going to make protein. And when she makes that protein, then now that protein can be used to assemble organisms or living things. That is the correct order for protein synthesis. Are there any questions? I know you do. I know you have questions. But unfortunately, I cannot answer. I hope the video has answered all your questions. Okay? Please subscribe to my video. And if you find it interesting, please do me a favor. Tell your friends. Tell your relatives. Pass the word out. This is Dr. Cisse. And I hope you have a very blessed day. And bye.